everyone, welcome to my channel. So today's topic of discussion is digestion of food. So we all know food enters into our body through the buccal cavity. The food enters in our body in the form of biomacromolecules. So those biomacromolecules are first broken down and then converted into simpler substances, then digested and absorbed. So we know the process of digestion is very complex and it is accomplished by the mechanical and chemical processes. So this line would be clear to you all. So the buccal cavity performs two major functions which we have to remember. The first function is the mastication of food and the second function is the facilitation of swallowing. In the buccal cavity, teeth and tongue are present. So the teeth and the tongue, with the help of the saliva, masticate and mix up the food thoroughly. Mucus in the saliva helps in lubrication and adhering the masticated food particles into a bolus. So the food is converted into bolus with the help of mucus that is present in the saliva. This mucus in the saliva helps in lubricating and adhering the masticated food particles, and that forms a bolus. Then the bolus is conveyed to the pharynx and then into the esophagus. First, the bolus enters into the pharynx, then enters into the esophagus by swallowing or deglutition. These both are synonyms. Either you call it deglutition or either you call it swallowing. The bolus further passes down through the esophagus by successive waves of muscular contractions called as peristalsis. Now, the food or the bolus enters into the esophagus by the successive waves of muscular contractions. These waves of muscular contractions are known as peristalsis. So, earlier I have told you that there is a gastroesophageal sphincter between the stomach and the esophagus. This gastroesophageal sphincter controls the passage of food into the stomach. So, the saliva that is secreted in the oral cavity contains electrolytes, that is sodium, potassium, chlorine and bicarbonate ions, and the enzymes salivary amylase and lysozyme. The composition of saliva is very important. Many of the times the question is framed on this part, that what are the constituents of saliva. So you have to remember these four electrolytes, enzymes and the salivary amylase, which is very important, and lysozyme also, as it is antibacterial in nature. So the chemical process of digestion is initiated in the oral cavity, as we all know, by the hydrolytic action of the carbohydrate splitting enzyme, that is the salivary amylase. This you have to remember. As we eat the food, the digestion and absorption is started from the oral cavity, and what helps in digestion and absorption there, that is the salivary amylase. About 30% of starch is hydrolyzed here by this enzyme. This you have to remember. What's the percentage of the starch that is hydrolyzed here? By this enzyme, the enzyme is salivary amylase. So that's 30%. Into the disaccharide and the maltose. So the starch is hydrolyzed here and it is converted in the disaccharides or the maltose. The pH at which this enzyme works appropriately, that pH is 6.8. This also you have to remember. So the lysozyme present in the saliva acts as antibacterial agent that prevents infections. This lysozyme is also present in our tears. This is also a very important question. You can note it down. So this starch is converted into maltose. This maltose is a disaccharide. So it is converted by the salivary amylase and the pH at which it is active, that is 6.8. Next, the food enters into the stomach. Now, the mucosa of the stomach has gastric glands. Stomach has gastric glands, and the gastric glands have three major types of cells. They are namely mucous neck cells, peptic or chief cells, and parietal or oxyntic cells. So what are the functions of these three cells which are present in the stomach? The mucous neck cells secrete mucus. The peptic or chief cells secrete Pro-Enzyme Pepsinogen. Parietal or oxyntic cells secrete HCl and the intrinsic factor. These both are important for the absorption of vitamin B12. So this can be asked. that what the axanthic cells or the parietal cells secrete. They secrete the HCl and the intrinsic factor. These are important for the absorption of which vitamin? Vitamin B12. Many times the question is framed on this part. Next comes the stomach stores the food for 4 to 5 hours. Food mixes thoroughly with the acidic gastric juice of the stomach by churning movements of its muscular wall, and that is known as chyme. So firstly we eat food. Food is converted into bolus. Then the bolus reaches the stomach. There occurs the churning movements with the gastric acidic juice, and the muscular walls churn it, and then chyme is formed. Next comes the Pro-Enzyme Pepsinogen. On exposure to the hydrochloric acid gets converted into the active enzyme pepsin. It's a very important part, that in which form we get this is the Pro-Enzyme Pepsinogen. When it gets the exposure of HCl in the stomach, that converts into the active enzyme pepsin, the proteolytic enzyme of the stomach. This pepsin is the proteolytic enzyme of the stomach. This pepsin converts proteins into proteoses and the peptones. These peptones are also known as peptides. It's an important part, that what's the function of pepsin? It converts the proteins into proteoses and the peptones. Next comes the mucus and the bicarbonates. These are present in the gastric juice, plays an important role in lubrication and protection of the mucosal epithelium from excoriation by the highly concentrated HCl. HCl provides the acidic pH. As I have told you, that salivary amylase is active under the pH of 6.8, and HCl provides the acidic pH, that is a pH of 1.8, that is optimal for the pepsins. So this question is also very important. At what pH the pepsins work appropriately? So the answer would be pH of 1.8. Renin is a proteolytic enzyme that is found in the gastric juice, and this is present only in infants. This renin is present in infants that helps in digestion of the milk proteins. Small amounts of lipases are also secreted by the gastric glands. These all you have to remember, that what the gastric glands secrete. Firstly, the pepsin, that is already converted from the Pro-Enzyme Pepsinogen. That is converted, for what it does, it converts the proteins into the proteoses and the peptones. Next, the mucus and bicarbonates are also secreted. What is the function of that? They help in lubrication and protection of the mucosal epithelium. Okay, next comes the renin. Renin is present in the gastric juice of infants. What it does, it helps in the digestion of milk proteins. Next comes the lipases, that is also secreted by the gastric glands. Next comes the various types of movements are generated by the muscularis layer of the small intestine. As I have told you, there are 4 layers. Here you can see there 4 layers are present. So outer layer was serosa, then comes the muscularis, then comes the submucosa and the mucosa, and I have already told you that what is present in the muscularis layer. So you can check out in the previous video. So here various types of movements are generated by the muscularis layer of the small intestine. These movements help in throughout mixing up of the food by various secretions in the intestine, and thereby facilitate the digestion. So as I have told you, that first the peristalsis occurs in the esophagus. Then there is a churning movements of the stomach that converts into chyme. And now finally in the small intestine there are various movements that is generated by the muscularis layer. So they also help in the mixing up of the food with various secretions in the intestine, thereby facilitate digestion. So what all are present? Bile is present, pancreatic juice is present, and the intestinal juice are secretions released into the small intestine. So what are the 3 parts of small intestine? I have already told you. The first part of small intestine was the duodenum. The second part of the small intestine was jejunum, and the third highly coiled part was ileum. So firstly, the food, the chyme from the stomach enters into the duodenum. So What else? What were the secretions that were entering into the duodenum? The bile, the pancreatic juice from the common hepatopancreatic duct, they were coming out. I can show you here. From the gallbladder, bile was coming out. From the liver, there was duct that was known as the hepatic duct. These both combine and form the common bile duct. And here, from the pancreas, pancreatic juice was coming, and that was forming a combined duct, and that was entering into the duodenum. So these all are the juices that are required for the digestion. So the pancreatic juice and the bile are released from the hepatopancreatic duct, as I have already shown you. The pancreatic juice contains inactive enzymes. Now these enzymes, you have to remember that what the pancreatic juice contains. The pancreatic juice contains the inactive enzymes, that is the trypsinogen, chymotrypsinogen, procarboxypeptidases, amylases, lipases, and nucleases. 1, 2, 3, 4, 5, and 6. These all you have to remember, all the 6 names of the inactive enzymes that are present in the pancreatic juice. So first, the trypsinogen is activated by the enzyme enterokinase, secreted by the intestinal mucosa, and that converts into the trypsin. So this trypsinogen converts into the trypsin, into the active trypsin with the help of the enterokinase enzyme, which in turn activates the other enzymes in the pancreatic juice. So firstly, the trypsinogen is converted into trypsin with the help of enterokinase. Now this trypsin further activates the other enzymes in the pancreatic juice. The bile released from the duodenum contains bile pigments. Now what are the bile pigments? These are the bilirubin and the biliverdin. Bile also contains the bile salts, cholesterol, and the phospholipids, but no enzymes. This is very, very important, that bile juice contains no enzymes. So what the bile does? Bile helps in the emulsification of fat. This point is very, very important. Now what is emulsification of fats? It helps in the breaking down of the fats into very small micelles. Bile also activates the lipases. The intestinal mucosal epithelium contains the goblet cells. Now what the goblet cells secrete? They secrete the mucus. The secretions of the brush board cells of the mucosa along with the secretions of the goblet cells constitute the intestinal juice or the succus entericus. This is very, very important, that what are the constituents of the intestinal juice or the succus entericus? They are the secretions of the brush border cells of the mucosa along with the secretion of the goblet cells. This name is also important, that what is the other name of the intestinal juice? It is the succus entericus. The juice contains a variety of enzymes like the disaccharides, that is the maltase, dipeptidases, lipases, nucleases, etc. Next comes the mucus along with the bicarbonate from the pancreas protects the intestinal mucosa from acid as well as provide an alkaline medium, that is of the pH 7.8 for enzymatic activities. So the submucosal glands, that are the Brunner's glands, also help in this. So the Brunner's gland are present. They are the submucosal glands which are present in the intestine. They also help in this process. Next comes the proteins, proteoses, and the peptones. What are the peptones? These are the partially hydrolyzed proteins in the chyme reaching the intestine are acted upon by the proteolytic enzymes of the pancreatic juice. They are given below. So this is all what happens in the small intestine. What happens to the proteins, peptones, and peptoses? As I have already told you, proteins, peptoses, and the peptones in the chyme reaching the intestine. When they reach the intestine, they are directly acted upon by the proteolytic enzymes. They are namely the trypsin, chymotrypsin, and the carboxypeptidases. And then these three are converted into the dipeptides. So this was all about proteins, and what happens to the proteins in the small intestine. Next comes the carbohydrates. When the carbohydrates in the chyme reach the small intestine, They are hydrolyzed by the pancreatic amylase, and they are also converted into the disaccharides. So that's very important. The polysaccharide or starch, it is acted upon by the amylase and converts into the disaccharides. Next comes the fats. The fats are broken down by the lipases first with the help of bile. Lipases are present in the bile. The fats are emulsified and then converted into diglycerides, and then these diglycerides are then converted into the monoglycerides that were the small micelles. This was all about fats. Now comes about the nucleases. The nucleases in the pancreatic juice acts on the nucleic acids. How the nucleic acids are digested. These nucleases are present in the pancreatic juice. These nucleases acts on the nucleic acids to form the nucleotides and then the nucleosides. First of all, these nucleic acids are present. Nucleases acts on it. It converts into the nucleotides, then converts into the nucleosides. Now, the enzymes present in the intestinal juice, all the succus and entericus, they act on the end products of the above reaction. These like dipeptides are formed, disaccharides are formed, monoglycerides are formed, and nucleosides are formed. The enzymes that were present in the intestinal juice, they act on the end products of these reactions to form the respective simple absorbable forms that are easily absorbed. Now, these final steps in digestion occur very close to the mucosal epithelial cells in the intestine so that the absorption is easy. Here it is given the dipeptides are converted by the dipeptidases. These are converted into amino acids. Maltose is converted into glucose plus glucose by the maltase enzyme. Lactose is converted into glucose plus galactose, that is converted by the lactase enzyme. Sucrose is converted by the sucrase enzyme into the glucose plus fructose. Nucleotides are converted by nucleotidases into the nucleosides, and these nucleosides are converted by the nucleosidases into the sugar plus bases. Di and monoglycerides are converted by the lipases into the fatty acids and glycerol. The breakdown of the biomacromolecules mentioned above occurs in the duodenum region of the small intestine. These all reactions were occurring in the duodenum region of the small intestine. So the simple substances thus formed are absorbed in the jejunum. As I have already told you, the first part of small intestine was duodenum, and these all processes occurred in the duodenum. Broken down food is converted or shifted into the jejunum, and then these are absorbed, and further they are shifted to ileum regions of the small intestine, and then they are absorbed there. So the undigested and unabsorbed substances are passed on to the large intestine. So there is very less absorption in the large intestine. So no significant digestive activity occurs in the large intestine. So what are the functions of large intestine then? Large intestine helps in the absorption of some water, minerals, and certain drugs which are not absorbed earlier. Secretion of the mucus, which helps in adhering the waste that was undigested waste, these waste particles together, and the lubricating it for an easy passage. So this was the function of the large intestine. Next comes the undigested and unabsorbed substances called feces enter into the cecum of the large intestine through the ileocecal valve that I have already taught you earlier, which prevents the backflow of the fecal matter. So this ileocecal valve prevents the backflow of the fecal matter back into the small intestine from the large intestine. So it is temporarily stored in the rectum till defecation. So the activities of the gastrointestinal tract are under neural and hormonal control for proper coordination of different parts. The sight, smell, or presence of food in the oral cavity can stimulate the secretion of saliva that we all have experienced. So the gastric and the intestinal secretions are also similarly stimulated by the neural signals. It's very important. The muscular activities of different parts of the alimentary canal can also be moderated by the neural mechanism. both local and through the CNS. This is very important, then how it is all moderated. So the hormonal control of the secretion of the digestive juices is carried out by the local hormones produced by the gastric and the intestinal mucosa. So this was all about digestion of the products. Thanks for watching and please subscribe to my channel before leaving this channel.